Hello my dear students, welcome back to your own channel VA NEET Academy. So our previous class was on cell cycle, so today we will be moving to the next obvious topic, mitosis. So in previous class and cell cycle we have seen that the cell is getting ready to enter into the mitotic stage, that is the cell division. So interphase was the stage where the cell was getting ready, itself preparing itself to enter into the mitotic, right? So why do we actually need mitosis? Now have you imagined whenever you receive a cut, you get hurt, scratches or anything, you get a new skin developed, healing happens, healing of wound starts, isn't it? So this new skin develops because the cell undergoes mitosis, or else you can say the cell multiplies to form a new skin cell. Okay, so this is the reason why we need mitosis, and a beautiful process of mitosis where the cell first divides its nucleus and then divide into two daughter cells, receiving each cell receiving one nucleus. A beautiful process. Understand the concept and do not mug up things. Thank you students. Let's go and see what happens in mitosis. So let's start our today's class, students. So let's start with mitosis. Definition. Okay, definition. Mitosis is an equational, okay, it is an equational division. So what happens in this? In which one parent cell divides, to produce two daughter cells, clear? And these two daughter cells has, so with same chromosome number, as what? As the parent cell. So the definition is very clear, isn't it? What does it say? Mitosis is an equational division, okay, equational division in which one parent divides into two daughter cell, okay, and these two daughter cell has the same number of chromosome as the parent cell, okay. Now because the chromosome number is unchanged with the, here it says with the same chromosome number, isn't it? Because the chromosome number is unchanged, and this is the reason why it is called equational division. So mitosis is an equational division where one of the parent cell divides to produce two daughter cell with identical chromosome number identical as the parent cell, okay. So now let us see why do we require mitosis, or else in other words, what are the major functions of a mitosis? So the one of the great function of mitosis is during growth, okay. So during growth, all of us know, as in my yesterday's intro video I have told you, the zygote, one cell stage called zygote is formed by the fusion of a egg and a sperm, okay. So this one cell zygote multiplies into 2 cell into 4 cell into 5 cell, and multiple cell stage called as morula, okay. So after the morula is formed, the cell becomes an embryo, and this embryo transforms into a baby. So in a growth, mitosis plays a major role, okay. So one of the function is growth. Second function is repair. Now when do we require a repair function? Now during the process of healing of wounds. Whenever you are hurt, whenever there is a cut in your body, whenever there is a wound in our body, repair has to happen, and mitosis helps us do that. Okay, or else tissue repair. Tissue repair, for example, you might have seen nowadays people are very fond of gym and all that, and they often go through the muscle tear and wear. Okay, so this muscle, after tearing, it has to be healed, so tissue repair, and for this we require mitosis. Third is replacement. So mitosis has a role in replacement also. Replacement of what? Of old skin cells. Okay, so old skin cells get repaired. All of us know we often shed our old skins which is blackened, and then we are glowed with the new skins. Okay, so this requires the process of mitosis. Also blood cell replacement, because blood cell has a definite life cycle, isn't it? So blood cell replacement also need mitosis. Okay, and the fourth very important role of mitosis is genetic stability. Genetic stability in the sense that it helps maintain the same number of chromosome when it is converted into a daughter cell. Okay, yes. So let's move on to the topic, the today's topic, that is mitosis. Now before going into the mitosis, I want you to just revise the cell cycle that we saw yesterday. So cell cycle has interphase, okay, and then M phase, and this M phase is what we are going to see today, that is mitotic phase. M phase has G1 phase followed by S phase and then G2 phase. Okay, so these three makes together the interphase. Okay, so G1 and G2 phase are the preparatory phase. What happens in S phase? Remember what happens in S phase? There is the synthesis of chromosomes. Okay, synthesis of chromosomes, DNA, synthesis of DNA. Okay, so for example, if the chromosome number is 46 chromosome number and 46 DNA molecules present. Okay, so after the S phase is over, the chromosome number will be same, that is 46 chromosomes, but then DNA will be doubled, that is 92 DNA molecules. 92 DNA molecules. So that's the role of a synthetic phase in an interphase. Okay, so after all this process is over, so in G1 and G2 phase, the cell gets ready, they grow, they divide, the organelles multiply, etc., that we have seen yesterday. Okay, so they prepare themselves to enter into the M phase. Now today we are going to talk in detail about what is M phase. Okay, right? So M phase has two events. Okay, so M phase in turn has two events. One is karyokinesis, okay, the other is cytokinesis. Karyokinesis and cytokinesis. What is karyokinesis? Karyokinesis is nothing but division of nucleus. Okay, cytokinesis is division of cytoplasm. So yesterday we have seen this topic also, right? Division of cytoplasm is cytokinesis. Karyokinesis is division of, karyo means nucleus. Karyo means nucleus. So division of nucleus. Cyto means cytoplasm. Division of cytoplasm. Okay. So M phase consists of two events, karyokinesis and cytokinesis. Now this karyokinesis in turn is divided into four stages: P, M, A and T. Okay, P, M, A and T. P is prophase. Okay, M is metaphase, A is anaphase and T is telophase. Okay, so karyokinesis is divided into PMAT. Remember the easy trick to remember, PMAT, PMAT, Prophase, Metaphase, Anaphase and Telophase. So, we will see this in detail now, what are these stages. Okay. So, first we will see what happens in prophase. The first stage is prophase. Okay. So, what happens in prophase? So, everything starts from prophase. Okay. So, the first thing which happens in prophase is the chromatin condenses. Okay, condensation of chromatin starts, chromosomes becomes visible, chromosomes visible. Why is it visible? Because the condensation happens. Okay, right? The nuclear, I am writing in short, nuclear membrane, NM is nuclear membrane. Nuclear membrane disappears. Okay, nucleolus disappears. Nucleolus disappears, right? And then there is the formation of spindle fiber. Formation of spindle fiber formation. So, these are the stages, or these are the events which happens in the prophase stage. Okay. So, for example, if I draw the cell like this, now this is your interphase cell, which has the thread like chromatin structure, okay, which has the thread like chromatin structure, and the nucleolus, which is a darkly stained structure, and this is nucleus. Okay, nucleus having the thread like chromatin, the cell. This is what stage? This is your interphase. So, during interphase, all the preparatory work happens, and now the cell is entering into the prophase. So, prophase, what did I tell you? The nuclear membrane disappears, and this is why I am drawing it in a dotted line, as a dotted line. And then there is the condensation of chromatins, and this is why there is a visible chromatin. There is a visible chromatin, and also the nucleolus has disappeared. So, I am not drawing the nucleolus there, and there is the formation of spindle fibers. There is the formation of spindle fibers. Okay, the formation of spindle fibers begins, right? So, this is in your prophase. This is your prophase. Okay, students. Now, what happens in late prophase? You can see during late prophase. Okay, I am drawing the sister chromatids in the late prophase stage. Let us make it more visible in the late prophase. So, you can see the sister chromatids. Okay. So, sister chromatids are there. Okay. Spindle fiber formation is there. Spindle fiber formation, and the spindle fiber is, sorry, this is another spindle fibers. This is this end. Okay. So, spindle fiber arises from the centrosome. Now, this is a centrosome, from where the spindle fiber arises. So, this is a centrosome from where the spindle fiber arises. Okay. So, this is the chromatids, and you can see I have drawn it as a sister chromatids. I have shown the sister chromatids. Now, where do we get the sister chromatids from? Because in the synthetic phase, in the interphase, we have seen that the chromosome, the DNA molecule has doubled, but not the chromosome. So, DNA molecule has doubled. I will draw better in a red. DNA molecule has doubled, but not the chromosome. So, this is your DNA molecule which has doubled. Okay. So, this is the sister chromatids. Clear students? Hope it's clear. Okay, so you can say this as a late prophase stage, late prophase, or else prometaphase. Prometaphase, that is just before the metaphase stage. Clear? So this is what happens in a prophase. Now next we will see what happens in a metaphase. After prophase, the phase is metaphase. So what happens in a metaphase? So in metaphase, chromosome arrange at the equator. Chromosome arrange at the equator. Okay. So or else you can say equatorial plate of the cell. And then spindle fiber, the next very important event which happens is spindle fiber attach to the centromere. To the centromere. What structure is centromere? Remember, centromere is the middle portion of a chromatids. Okay, centromere is the middle portion of a chromatids. Okay, so one of the NEET based question, very often NEET based interesting fact is that the best stage for chromosome study is this metaphase. Okay, why? Because the chromosome condensation has already happened, and this is the best stage where the chromosome can be studied. And prophase is said to be the most longest phase in the mitotic stage. So these are the important NEET facts often asked in the NEET question paper. Okay, so we will see how does a metaphase stage cell looks like. So in a metaphase, what happens? The chromosomes arrange in the equator. Okay, they arrange in the equator. Now because we have a sister chromatid, I am drawing the sister chromatids as well. Okay, and next what happens? Next important stage is the spindle fiber attached to the centromere. Now this is the centromere region. Okay, so spindle fiber attached to the centromere of a chromosomes, chromatids. Okay, clear students? So this is what stage? This is your metaphase. So in metaphase you can see the chromatids are arranged at the equator of the cell. Okay, so next phase is your anaphase. Next phase is anaphase. So what happens in anaphase? Most important stage in mitosis. Here the centromere splits. The centromere splits. Most important stage in anaphase, mitosis. Centromere splits and the sister chromatids they move to the opposite pole. They move to the opposite poles. Now this is what happens in a anaphase, a very important stage. Okay, so if you try to draw and one of the important NEET question is that centromere split happens during anaphase. Centromere split happens during anaphase. So let us try and draw the anaphase stage. So cell, now what did I tell you? The chromatids split. Now why does the chromatid split? Because the centromere splits. Okay, so there is the splitting of the centromere. So here I will show it in a, so this is your sister chromatids, right? Okay, so this sister chromatids are split because the centromere splits. Okay, and this is attached with the spindle fiber, and the spindle fiber pulls these sister chromatids to the opposite pole. So this bunch of chromatids will be moving towards this end, this direction, and this bunch of chromatids, sister chromatids, will be moving towards this direction. Okay. So this is what stage? Anaphase. This is your anaphase. Hope it's clear. Okay. So right. So last stage is your telophase. So telophase, what happens? Telophase. So in telophase, the major events which take place is chromosome reach to the pole. Chromosome reaches to the pole. Okay, chromosome reaches to the pole, and then the chromatids, or else you can say chromosomes, decondense. That means decondense. So it will be looking like a interphase chromosome, like thread like structure will reappear. Okay. So nuclear membrane, what happens to the nuclear membrane? It reappears. And what else will reappear? Nucleolus, which was gone during the prophase, will reappear. Okay, nucleolus will reappear, right? So what is the end result? End result is the formation of two daughter nucleus. Two daughter nucleus. So this is the end result of a telophase. Two daughter nuclei are formed. Two daughter nuclei are formed. So let us try and draw the anaphase stage chromosomes, anaphase stage cell. So the cell is here. Okay. So there is the reappearance of the nuclear membrane. So the reappearance of nuclear membrane, and it has moved towards the pole. Okay. So one has received the chromatins, decondensation happens, so thread like structure is formed, and nucleolus reappear, which is a darkly stained structure. Okay. So here also the chromatin decondensation happens and the nucleolus reappear. Clear? So this is how you can say, and the nuclear membrane is reappearing, so kind of forming. So this is the two nucleus present in one cell. Now the cell is just one, but then the cell has received two daughter nuclei. Now what stage is this? This is your, after anaphase, telophase. Okay. So after anaphase, the telophase. So one of the events of mitosis is over. So I told you that mitotic phase has two events. Karyokinesis, which is over, that is PMAT has taken place already. Now this next stage is cytokinesis. What is cytokinesis? Division of a cytoplasm. Now if you remember my yesterday's class, division of cytoplasm is nothing but, in animal cell it is the formation of cleavage furrow. Okay. Please do refer to the previous video, long video, not the MCQs, the long video where I have talked about the division of division of cytoplasm, or else cytokinesis. So animal cell undergo cleavage furrow, giving rise to two daughter cells with equal amount of chromosome nucleus. Okay. And in plant cell, what happens? Plant cell, there is a formation of cell plate formation. Cell plate formation, which in turn forms gets converted into cell wall, and this is how the plant cell gets converted into two daughter cell. Okay. If you remember yesterday's class, Okay. So, two daughter cell with cell plates, and this is the cell wall. I'm sorry, I was not showing on camera. Yeah, so this is how the two daughter plant cell is formed. Please do refer to the previous upload of my long video. I think it is on cell cycle. Okay, please do refer for the detailed diagrammatic explanation of cytokinesis. So hope the class was clear, student, and do comment and ask me if you have any doubts, and do subscribe, follow, like and share our channel, and this will really motivate me to bring you more and more videos. Thank you, students.